What are the chemical and biological or bioelectric mechanisms of methylene blue in preventing or treating neuronal degenerescences? Methylene blue, MB, also known as methylthioninium chloride, is a well-known synthetic dye and medication that has been reproposed for its potential neuroprotective effects in neurodegenerative diseases such as Alzheimer's disease, AD, Parkinson's disease, PD, and other topathies. Its mechanisms of action are multifaceted, involving chemical, molecular, and cellular pathways. Below is a detailed explanation of its mechanisms in the context of neuronal degeneration. 1. Inhibition of tau protein aggregation. Tau pathology. In neurodegenerative diseases like Alzheimer's, hyperphosphorylated tau proteins form neurofibrillary tangles, NFTs, which disrupt neuronal function and lead to cell death. MB's role. MB acts as a tau aggregation inhibitor. It binds to tau proteins, preventing their misfolding and aggregation into toxic oligomers and fibrils. This is achieved through redox chemistry. MB's ability to accept and donate electrons allows it to interact with cysteine residues in tau, stabilizing its structure. Disruption of sheet formation. MB interferes with the formation of sheet structures, which are critical for tau aggregation. 2. Mitochondrial function enhancement. Mitochondrial dysfunction. Neurodegenerative diseases are often associated with impaired mitochondrial function, leading to reduced ATP production and increased oxidative stress. MB's role. MB acts as an alternative electron carrier in the mitochondrial electron transport chain, etc. Cytochrome C reductase activity. MB can accept electrons from an ADH and transfer them to cytochrome C, bypassing complex IL-3 dysfunction. This enhances ATP production and reduces oxidative stress. Antioxidant properties. MB scavenges reactive oxygen species, ROS, and reactive nitrogen species, RNS, protecting neurons from oxidative damage. 3. Modulation of autophagy and lysosomal function. Impaired autophagy. In neurodegenerative diseases, dysfunctional autophagy leads to the accumulation of toxic protein aggregates and damaged organelles. MB's role. MB enhances autophagy, promoting the clearance of protein aggregates like tau and synuclein. Activation of TFEB. MB upregulates transcription factor EB, TFEB, a master regulator of lysosomal biogenesis and autophagy. Lysosomal acidification. MB improves lysosomal pH, enhancing the activity of lysosomal enzymes that degrade misfolded proteins. 4. Anti-amyloid effects. Amyloid A. Pathology. In Alzheimer's disease, plaques contribute to neuronal toxicity and inflammation. MB's role. MB inhibits the aggregation of apeptides and promotes their clearance. Binding to A oligomers. MB binds to A oligomers, preventing their aggregation into fibrils. Reduction of A production. MB may modulate the activity of beta and secretases, enzymes involved in A production. 5. Anti-inflammatory effects. Neuroinflammation. Chronic inflammation mediated by microglia and astrocytes exacerbates neuronal damage in neurodegenerative diseases. MB's role. MB reduces neuroinflammation by inhibition of NFB pathway. MB suppresses the nuclear factor kappa B, NFB, pathway, reducing the production of pro-inflammatory cytokines. Modulation of microglial activation. MB shifts microglia from a pro-inflammatory, M1, to an anti-inflammatory, M2, phenotype. 6. Neurotransmitter modulation. Neurotransmitter dysregulation. Neurodegenerative diseases often involve imbalances in neurotransmitters such as acetylcholine, dopamine, and glutamate. MB's role. MB modulates neurotransmitter systems. Cholinesterase inhibition. MB inhibits acetylcholinesterase, ACE, and butyrylcholinesterase, BUTCH, increasing acetylcholine levels and improving cognitive function. Dopaminergic protection. In Parkinson's disease, MB protects dopaminergic neurons by reducing oxidative stress and mitochondrial dysfunction. 7. Epigenetic regulation. Epigenetic changes. Neurodegenerative diseases are associated with aberrant DNA methylation and histone modifications. MB's role. MB influences epigenetic mechanisms. Histone demethylase inhibition. MB inhibits lysine-specific demethylase 1, LSD1, leading to changes in gene expression that promote neuronal survival. DNA methylation modulation. MB may alter DNA methylation patterns, restoring normal gene expression in neurons. 8. Neurogenesis and synaptic plasticity. Impaired neurogenesis. Neurodegenerative diseases are characterized by reduced neurogenesis and synaptic loss. MB's role. MB promotes neurogenesis and synaptic plasticity. BDNF upregulation. MB increases brain-derived neurotrophic factor, BDNF, levels, supporting neuronal survival and synaptic function. Synaptogenesis. MB enhances the formation of new synapses, improving neuronal connectivity. 9. Redox cycling and antioxidant properties. Oxidative stress. Excessive ROS production is a hallmark of neurodegenerative diseases. MB's role. MB acts as a redox cycler. At low concentrations, MB functions as an antioxidant, scavenging ROS and protecting neurons. At high concentrations, MB can paradoxically generate ROS, which may have therapeutic effects in certain contexts by inducing selective toxicity in damaged cells. Clinical implications and challenges. Dose-dependent effects. MB's effects are highly dose-dependent, with low doses being neuroprotective and high doses potentially toxic. Blood-brain barrier penetration. MB readily crosses the blood-brain barrier, making it an attractive candidate for treating central nervous system disorders. Clinical trials. MB has shown promise in early-phase clinical trials for Alzheimer's and other neurodegenerative diseases, but larger studies are needed to confirm its efficacy and safety. Conclusion. Methylene Blue's multifaceted mechanisms of action make it a promising therapeutic agent for neurodegenerative diseases. Its ability to target multiple pathological pathways, tau aggregation, mitochondrial dysfunction, oxidative stress, inflammation, and synaptic plasticity, highlights its potential as a disease-modifying treatment. However, further research is needed to optimize dosing, minimize side effects, and fully elucidate its long-term benefits.